we talked about just a little introduction to genetics and chromosomes and chromosome number. And then we talked a little bit about Gregor Mendel. Remember the Austrian monk who did his research with what?
Pea plants.
Pea plants. Grew thousands and thousands of pea plants. Carefully recording his data, carefully cross-pollinating from one plant to another, growing the resulting seeds and seeing what the results were. Categorizing different traits and characteristics and looking at the numbers. That's what Mendel did. Okay, for example, he may have taken a pea plant with purple flowers, a pure purple flower pea plant. And he'd take a tiny grain of pollen, or several grains of pollen with a paintbrush. Okay? And what he would do is he would transfer the pollen, okay, to the stigma of a female flower, allow it to be fertilized, allow the fruit to develop and grow into seeds. Then he'd take those seeds and plant them. And when he grew, he'd look, okay, well, what is the flower color of this offspring? And record those numbers, record that data. So he's very careful. He was looking at a bunch of different characteristics, 7 or 8 characteristics about those pea plants. And like we said yesterday, he started to recognize some patterns in that. He started to recognize that given certain parents, he always saw the same ratios in the offspring. And that's what led him to his conclusions. So Mendel didn't know anything about genes. This is well before genes and DNA had been discovered. Okay? And so his work was basically statistical work, looking at the numbers. So like we talked about in our question today, if we're thinking about humans, in human cells, there's 46 chromosomes. Okay, they come in pairs, okay, 23 of each pair. A sperm cell gets 23 chromosomes, the egg cell gets 23, and the resulting embryo ends up with 46 chromosomes. So we will use this word genes a lot. Okay? And Mendel didn't know about genes. Like I said, he didn't know about DNA. He called these factors. Okay? There's factors from the parents that contribute to the offspring. Okay? And what Mendel's research showed is that the genes, we might call them alleles, but we'll generally call them genes, come in sort of 2 forms. For what he was looking at in his pea plants for these characteristics, what he said is that the genes come in 2 forms. One form is the dominant version, and one is the recessive version. And so what he meant by that, the dominant version of a trait, you could think of it as kind of like the stronger form. It kind of shows through whenever it's present. The recessive version, you can think of it as kind of the weaker form, that it can be covered up, it can be hidden by the dominant version if they're both present. When we are talking about genetics, we will usually use letters as symbols for these different genes. And when we're writing the symbol for a dominant gene, we use a capital letter. For example, if we're talking about the gene for a tall pea plant, we would use capital T, T for tall. When we're talking about the genes, the symbols for a recessive version, we use a lowercase letter. For example, in a short pea plant, because we're talking about sort of 2 aspects of the same characteristic, we would use a lowercase t, okay? Because it's the recessive version. Okay? So we use the same letter we used for the dominant trait, but we use it as a lowercase letter. So if we imagine the pea plants, each pea plant has two genes that determine its height. One came from each parent. And we have some words. Sometimes the organism can be what we call homozygous dominant. Homozygous means it has two of the same genes. And saying homozygous dominant means they're both the dominant gene. So big T would be homozygous dominant. Or a plant could have two of the short genes. And we would call it homozygous recessive. So, this pea plant that had two big T genes, two dominant genes, would it be tall or short?
Tall.
Tall. It has two tall genes, it would be a tall pea plant. How about this one? The homozygous recessive plant? Short. Well, there's one option we haven't gotten to yet. What is it?
One's tall, one's short.
Yeah, a tall gene and a short gene. There's a different word for that. That's called heterozygous. It means one of each. So a big T and a little t. So what would that plant look like?
It could be short or tall.
Nope.
Short.
Middle, what's more powerful?
So most of the time people will say, well, it'll probably be in between. Has a tall gene and a short gene. It's probably somewhere in between. But in these characteristics that Mendel was looking at in these pea plants, there wasn't really an in-between. Pea plants were either tall or short. Okay? Which would it be?
Tall.
Why?
Because it's dominant.
Right, because it has one of those dominant genes. The tall is the dominant version of height in pea plants. And so it has the dominant trait, has the dominant gene. That's what shows through. The recessive version is kind of covered up, you could think of it, by having just one dominant gene. So yeah, that's a tall pea plant. All right. So we have some terms we need to use when we're talking about genetics and looking at these characteristics. We can talk about an organism's phenotype and its genotype. When we talk about an organism's phenotype, we're talking about how those genes sort of show through. What do they result in? Often, for our purposes, we're going to find what is its physical appearance? How do those genes translate into the actual organism? So for example, saying the plant is tall, that's its phenotype. Saying the person has blonde hair, that's a phenotype. We're talking about how the genes come through. But if we talk about an organism's genotype, we're talking about what actual genes it has. What letters does it have that make up and result in those traits? And these are letters. So when we're talking about genotypes, we're talking about letters. Big T, little t, big B, big B. Okay. Big S, little s. Homozygous dominant, heterozygous. Those are talking about the genotype of these organisms. So if we see, we have two pea plants here, tall and short. So what's the phenotype of this plant? Tall. Again, it's a word. It's how those genes come through. What are its possible genotypes? Raise your hand, please. Well, they're all T's.
Capital T.
Could be a big T, two dominant genes. It could be homozygous dominant. What other combination would result in the tall phenotype, Julia?
Small T.
Yeah, or it could be heterozygous. Either one will result in a tall pea plant. Tell me about the phenotype of this plant. Sophia? phenotype.
Oh,
It's short. Again, we're describing it. And Sophia, what are its possible genotype?
let me try and find recessive.
Right. What is the term we would use for its genotype? Two words actually. Homozygous recessive. Yes. Little t is homozygous recessive. Good. All right. Now, I guess I'll say this now because I tend to repeat this a lot. The traits Mendel was looking at in pea plants were very simple traits. Most of the ones he was looking at were just controlled by one set of genes. And there was a simple dominant and a recessive gene that resulted in tall plants or purple flowers or green peas. But in reality, with most organisms, genetics are much more complicated than we make it sound when we're doing these simple examples of Punnett squares. There's not a gene for height in humans. That if you get the big T, you're going to be tall. If you get the little t, you're going to be short. As you know, there's wide variety in heights. And there are in-between heights in humans. Okay? Didn't happen to be the case in pea plants, but that's because complex traits like height in humans or lots of other things are controlled by many different genes. It's not just one. But for our purposes, to sort of simplify, and this was true of Mendel's plants that he was looking at, they were relatively simple traits that were either dominant or recessive. So sometimes people say, well, what about in humans or this? And it's not as easy to sort of take these simpler examples and expand them to humans. But this will give us the basics about dominance and how we can predict offspring. So once we know the genotypes of parents, we can predict with certain probability what their offspring might be like.
I think I know what these are.
All right, good.
You write like the T's here and the T's
Yep.
there, and then you write in the box which one you
Yeah. So these are Punnett Square diagrams. Okay? We'll do simple ones that are just a single trait being crossed. And what they do is they help us predict what would happen if these two parents had offspring. And so what we do is you have to know the genotypes of the parents to begin. So I'm giving you one here. I'm saying, okay, what if one pea plant was homozygous dominant, had two big T's, and it produced offspring, it pollinated a plant that was homozygous recessive, two little T's. What would their offspring be like? Before we do this, could anyone just look at those parents and figure out what would the offspring be like, Meg?
Wouldn't it be tall because they're the two dominant?
Okay. Yes, and Vlad, what were you going to say?
Heterozygous,
How do you know that?
because you're crossing a tall plant and a short plant, which is has the genes of dominant, homozygous dominant and homozygous recessive.
Yeah, you're right as well. You're both right. So just before you go to the Punnett Square, when this plant produces pollen grains and the chromosomes get split up, what gene can this give to its reproductive cells? It can only give the big T, right? That's all it has. And how about the egg cell? What can it give? Just the small t. And so if you have one of those cells pollinate each other, that's only one possibility you end up with. The way we figure it out in the Punnett Square, we take one parent's genotype and we write it on the top of the Punnett Square. So let's take the homozygous dominant parent. Doesn't matter which side you use. We take the other parent's genotype and we put it on the left side of the Punnett Square. And then we combine to fill in each box. So this box is going to get what? What's it get from the top?
Oh.
Big T. And what's it get from over here? Little t. What's this box get from the top? And the side? Oops. That one, big T, little t. Big T, little t.
Yeah, wouldn't they all be there too?
Yeah, so like Vlad said, in this cross, they're all going to be heterozygous. Like Meg said, what are they going to look like, Meg? They're all going to be tall. And so what we often will do is we'll write in the percentages. So the possible gene, so each of these boxes represents an offspring, or 25% of the offspring. Okay. So, possible genotypes we end up with, or just one, heterozygous. 100% of them are in this are going to be heterozygous. Could this cross ever result in a homozygous recessive offspring?
No.
No. How about a homozygous dominant offspring?
Yes.
No. All of the offspring, the only possibility in this case, they're all going to be heterozygous. How about phenotypes? What? Tall. How many? What percent?
Oh, 100%.
Yeah, 100% of the offspring are going to be tall in this case. And Mendel saw this. What Mendel did is he had pure breeding tall plants, and he had pure breeding short pea plants. And whenever he cross-pollinated them and grew the seeds, they always produced tall plants. Okay? He never got any short pea plants from that cross. Okay, well that's not always the situation though. Let's look at another Punnett square. Now we have big T, little t. It's one parent. What would we call that?
Heterozygous.
Heterozygous parent crossed with somebody else. What would the little t parent be called? Hidden? Homozygous recessive, correct. So we put one parent on top, one parent on the side. Doesn't really matter which one goes where, as long as they stay together. All right. Karina, what's going to end up in this box? What's it get from the top? No, what letter does it get from the top of its Punnett square? Yes, what's it get from the side? So it's going to be heterozygous. Evan King, what is going to end up in this box down here?
Big T, little t.
Correct. Big T from the top, a little t from the side. Jackson, what's going to end up in this box?
Little t, little t.
Little t from the top, little t from the side. And Bella, how about this box?
Little t.
Correct. Okay, so we've completed our Punnett square. Okay, so what are the possible genotypes that we end up with as a result of this cross? Matt?
Big T, little t, 50%.
Yes? And?
Little t, 50%.
Correct. So 50% are heterozygous, 50% are homozygous recessive. What about phenotypes? Check.
So that'd be 50% tall, 50% short.
Correct. 50% of them are tall, 50% are going to be short. And let's do our last one. You guys go ahead and fill in this last one. You complete the Punnett square. And fill in the phenotypes and genotypes with the percentages. And then I'll walk around and look at it. They have to stay together. So it has to be big T, little t, doesn't matter if it goes here or here, but they have to stay together. Okay, good. Yep. Looks like you guys are doing pretty well. So we put one parent on top. Now a couple things. So the two letters for each parent need to stay together. So the big T and little t here need to stay together. It doesn't matter if you put them here or here, but they stay on the same side. And the other parent goes here. Fill in our Punnett square. And we end up with our genotypes. And what percent are big T, homozygous dominant? Evan Ziegel?
T is 25%.
They're all T's.
Big T, 25%.
Yes, the homozygous dominant, 25%. And I guess I figured the rest. The heterozygous is 50%, and the homozygous recessive is 25%. Again, Mendel always saw this combination. Whether it was the color of the flowers, the color of the peas, the texture of the peas, when he crossed two, he called them hybrids, two heterozygous plants, always ended up with the same ratio of 25% to 50% to 25%. Now, well, let's finish this. Hold on one second. Phenotypes. What percent would be tall, Kaden? 75% tall, and obviously 25% short. Now, these are probabilities. And as we know for probabilities, they don't work out perfectly. For example, I've flipped a coin at lunch for the last 5 times or so that we've gone to the gym. And I've lost all of those flips. Now, probability would say I should have won about half of them, but I didn't, because probabilities are based on chance, and they don't always work out. So could these two plants produce 10 seeds, and they're all tall?
Yeah.
It's possible. Okay? It would be against the probability a little bit, but it's possible. Just like I could flip a coin 5 times and not get a single tail. It's possible. Okay? But they are probabilities. So in large numbers, they work out pretty well.
When you're writing something with a capital T and a lowercase t, does the capital always go first?
Yeah, we usually, by convention, it doesn't matter, but we put the capital first.
Yeah, that was.
Yeah, we always write the capital letter first. We also have to be careful, especially in the letters you're using. Sometimes it's easy to make the letters look similar. For example, like the letter, I'm trying to think, sometimes F, C, yeah, like, so what I'll often do, like, because obviously capital C, lowercase c look the same, what I often do, I'll make the uppercase one print, and I'll make the lowercase one like a cursive lowercase c. Or an F, like, I'll make very clear that my capitals versus my lowercase, it's just easier to make it. And really the letter, it doesn't, we use as convention the first letter of the dominant trait, but really the letters don't matter as much, as long as you understand what you're talking about. All right, so you guys understand the Punnett Squares thing?
Yes.
All right, let's do this one real quick. Now let's look at a different trait. Peas come in 2 colors, yellow versus green. Okay? Like it says here, yellow is the dominant, we would say allele, the dominant version. So what if I cross what's the word we would use for this genotype? Homozygous dominant. This one is homozygous recessive. Do the Punnett Square showing what would happen if those 2 pea plants produce some little baby pea plants. And then answer those 2 questions. Yeah, when you're doing it, make like super little tiny little y's and really big capital Y's, so you're really clear about what the difference is.
Is that clear enough?
That's pretty clear. All right. One parent goes on top. The homozygous dominant in this case, doesn't matter. Could put the homozygous recessive one there. You'll end up with the same result. The other parent goes on the side. Fill in each box. What percent are yellow? 100% yellow. 0% green. Got it? Anyone have any questions about how we complete a Punnett Square? All right. So that's good. So what I have for you